I'm Kenny Pacharka. I work with high-tech seed production here out of Southern Alberta. Our farm's just north of Coaldale on the 845, and I've been working here in the industry for 7 years now. So a lot of the stuff I'll be reviewing in the history program was a little bit before my time, but nonetheless been involved in some of the big changes and advancements that have been made.
I'll introduce myself.
Sure. Sounds good. So to start, who here currently grows canola commercially or hybrid seed production? So we got a few people. Right on. Well, some of this early production might apply to you, and whether it's the new stuff or not, just feel free to ask questions, ask for clarifications, anything like that as we go along. We'll be covering some of the history pre-hybrid, so kind of the period up till the late 80s, early 90s when the hybrid started to ramp up, and then into the changeover into hybrid programs, intensive breeding, high investment, and ultimately where we're at today in the industry. So before hybrids, just to give you an example of the speed of change at that time, in a period of 44 years until 1984, there were only 24 new canola or rapeseed varieties registered. This is quite slow. Main reasons for this would be it was mainly done by public breeding programs. The development was slow because seed sales volumes were a lot lower. Commercial production was a lot lower. Canola or rapeseed oil was not as big of a market at that time. The production was done by your CSGA seed growers, production and retail. Some of the companies who've made up the current big names you see out there were involved in production, but a lot of CSGA growers were producing and retailing. And seed production was on dryland acres. It wasn't being done in Southern Alberta. And at the same time, sales volumes were low. Farmers would reuse seed. There was a lot of bin run seed used. It wasn't patented and protected the way it is here today. So that was. Yeah. So it was a very different environment. But yeah, the products, they were OPs. They were open pollinated rapeseed varieties of either Polish canola, which we were lucky enough to have a plot here. We wouldn't see a lot of those plots around the last few years, but the Polish canola, being a short season variety, is back on the map here, and some canola varieties as well. Actually, the wind quieted down. I don't think I'll need the clip. In the 1980s, a shift came about, and that was a move towards biotechnology, plant breeding, genetic systems. Really, there was a lot of investment in anything biotech at that time. So money was being pumped in. The private sector was moving into the seed industry, not just canola. Canola is one of the crops to benefit, but your corn, your soybean, your cotton, your sunflower, kind of hybrids started to become the way. So you had the private companies investing, and at the same time you still had the public companies working on the genetic systems as well. And in canola specifically, the focuses were heterosis or hybrid vigor, which is the crossing of two genetically different parents to create a hybrid product that has increased performance over both parents. So hybrid vigor was the goal, and the hybrid systems were systems where you used male sterility. You would have a pollinator and a sterile male and cross them. So the sterile male could not pollinate itself. The foreign pollen comes to that plant, pollinates it, and you get your hybrid. That's the idea there, but I'll leave that up to the expert here in the next talk. But a big date that would lead to a lot of the basically patents on the varieties was in 1990, plant breeders' rights legislation was passed. So the breeders could basically patent the varieties, have control over their production, and anyone who wanted to use them then had to pay a fee, the seed cost, the technology use, those kinds of things. So that started in 1990. So like I touched on briefly before, you're basically, to create a hybrid, you're taking two unrelated parents, you're crossing them through bee pollination in the case of canola, and you're creating F1 progeny. And the goal is that the progeny will outperform both the parents, create the hybrid vigor. Challenges on the production end, which is what you see in Southern Alberta, is that you need to find parents that combine well. So the breeders take as many different canola genetics that they can and plant all these plants together, male and female crosses, evaluate them for a number of generations to see what hybrid produces the best yield, the best oil profiles, maturity, height. There are a number of characteristics they're targeting in the breeding. Then they need to, once they find the hybrids that are the best, they need to send them out to production. And you need to be able to produce the seed in a cost-effective manner. You need to be able to hit appropriate yield, that the cost isn't through the roof. You need consistent quality, just security on the investment that's being made at that level. So now we'll touch on the first hybrids that were registered. And the first hybrids came from a student at the University of Guelph had a project. His name was Paul Banks, and he started working with a male incompatibility system, was what his system was called. And it led to two hybrids coming out in 1989, one by King Agro and one by Allelix, the KC701 and HCN1. They had some issues that led them to not be marketed commercially, and that was you couldn't get the cross to set an efficient seed to make it cost effective. You just couldn't produce the yield that made the cost of seed something a farmer would buy. So instead of, if it's $8-9 a pound, it would have had to been much higher, and there's no way the farmer was going to be buying it. So those did not end up as commercial successes. The first successes, though, came from the Polima CMS system, and this system was developed by Pacific Seeds in Australia, a gentleman by the name of Greg Buzza, and Pacific Seeds also had a sister company in Canada, Zeneca Seeds. And so when they created these spring hybrids for the Canadian market, they marketed them through Zeneca, and Hyola 40 in 1989 was their first variety. It was a good hybrid, but the problem was in the production of it. They couldn't get effective pollination. And afterwards, if we have time, we can look at the flowers in the canola. But their issue was that the flowers were very open. They weren't closed. And so when the bees went to forage on the nectar, they could enter from the side, and they would completely miss the pistil on the top of the plant. So when they missed the pistil, they weren't dropping the pollen in, and they weren't getting pollination. So that was the challenge with that hybrid, and it did not achieve commercial success. But the Hyola 401 in 1991 did. It was a great product. It was short, had good standability, high seed size, high yield. It just outperformed the OPs drastically, and that was a big product for quite some time for them. It eventually became Hyola 357 once the Roundup Ready gene was bred into the product, but that was the first big success. That product actually survived a long time as far as Currentday hybrids are concerned. It was until blackleg resistance was mandatory that product was put on the shelf. After this system, the MSL Lemke system is out of Germany. Werner Paulmann created that, and it was originally for winter canola hybrids. And it's created some of the best hybrids in the winter European market, and eventually they came to North America, mainly in the US winter canola market as well. Baldur and Kronos are those varieties. But eventually it did come to Canada through Agri-Progress Incorporated. Otmar Philipp was the gentleman who brought it over on a partnership with NPZ, and these hybrids have been top performing hybrids in the Clearfield and Roundup Ready segment. Up till today, DL Seeds currently breeds these hybrids, and they market them through various other Canadian distributors who pick up the varieties. The Ogura system is from INRA. It's the national research group in France. And in the early 1990s, Allelix picked up rights to breed with this genetic system. They are Pioneer Hybrid now. They soon after became Pioneer Hybrid. And they found this system in radish, which is a similar plant to canola. It's another brassica plant, and they found it in that and converted it into, or transferred it into canola. Took a while to make the system functional to meet human consumption standards because of high glucosinolates, which are toxic to the body. But once they bred those out, in 2001 they released their first hybrid, 45H21, and they're still introducing top Roundup Ready hybrids on the system today. A number of other companies have also taken on this system. DSV is a big one in the late 90s and into the 2000s. They now work with DL Seeds as well here in Canada. Cargill picked it up in 2000, and their specialty canola oils are produced on this system. And so are Monsanto. After they bought Advanta in 2004, they started, and Dow in 2010 introduced their first Ogura hybrids after their long run of OP production. So the other main male sterility system out there is the Barney's Barstar, which eventually became Invigor. So this is Bayer's brand. It was originally from Plant Genetic Systems out of Belgium, and they came to Canada with the system for the purpose of getting into the spring canola industry. They saw it as a big opportunity, and it was the first transgenic male sterility system, and they also bred in selection resistance to the Liberty herbicide. So that's what they used to select for the hybrid in that crop. And their first hybrids came out in 1996, 3850, 3880. Eventually, this company became Agrivo, and eventually there was a merger with them and Roan Poulenc to become Bayer Crop Science in 2002. And they're the largest seller of spring hybrid canola in the world. So they're a very, very big player in the Canadian market.
You said they sold 1 million bags this year.
Yeah, I believe it. It's for the market. I don't know if anyone's heard the numbers finally, how many acres there are this year, but I'm sure 16, 17 million. It would have been higher if a lot of that area in Saskatchewan and Manitoba would have got seeded, but they're the...
Scan posted 19.5, and then there was a big kerfuffle that they don't know what they're talking about.
I wouldn't surprise me that was the planned acres,
Yeah.
but if it was that high seeded, that would be pretty wild. So, ultimately what we're here to talk about is the seed production in Southern Alberta. What you see around you, what you see right here is an example, but when you drive down the road, you see the tents, you see the crop in bays, the small males, the big females, beehives in the corners, all that kind of thing. The reason it came to Southern Alberta, there are a number of critical success factors basically that had to be met because of the investment costs in the breeding, and the genetics, parent seed production, and finally the hybrid seed production that you see. And these factors were water availability. So the irrigation, pivot irrigation in Southern Alberta is vast, and it supplied the requirements of the industry early on and still today. It's adequate to provide the needs, whereas other areas in Canada don't present the same amount of irrigated land. There were established beekeepers, which are important because you need the pollination. You need to have bees at the field or you don't get any seed production. So there were good beekeepers established then, and there are a lot of beekeepers now to meet industry needs. Disease pressure is low. It was considered blackleg free in the 90s when production started to come here. And even today, with the generally dry and hot climate, we haven't quite seen it the last few years, it's less pressure from sclerotinia and other diseases that affect canola. They are definitely increasing as canola increases, but rotation and isolation are huge for what we do. We require a mile of isolation from any other canola, because that's a reasonable distance that the bees will be focused on the flower in our fields that we place them around. Anything closer, you start to get more bee foraging outside, and that ultimately leads to problems with hybridity and a product that you cannot sell. And length of the growing season. We can generally seed earlier and harvest later with less risk of a hard frost. So you go further north and further east, you don't have that same benefit. So, to sum it up, out of Canada's market, if it's 19.5 or if it's 17 million acres, just over 2/3 of that seed is produced here in Southern Alberta. So that's a lot of bags of seed that are produced in one small area. The rest of it would be, there's a little bit in Saskatchewan, there's some in the US, and then Chile is a pretty big contra-season production area for most companies are producing there, whether it be on the breeder, parent seed, or on certified production. We ourselves produce there as well. And what you see outside of the fields is you see plants such as that Pioneer plant right there, Monsanto down the road, Bayer down the road, ourselves are here, Dow's here, so you see production programs being put in place with people, infrastructure, as well as a lot of seed processing investment has been made because of this production. So here's just a couple little looks of where things have gone. I don't know if you want to come closer or if you want to come take a look after, if it has interest to you. But these numbers are from our friends at the Canola Council. And it just shows where the market's gone since '86. Really the hybrids started to gear up early 90s, and really in the 2000s have taken over. And now would be more than 95% of the seed sold is hybrid seed. So you just see this top dark bar is the number of acres that of commercial canola that went in. So we were just over 6,000, 6,586, and now we're up over 16,000 for the last couple, 2-3 years. Or 16 million, sorry, let's speak in the right terms here. And if it is 19.5, then we've got one of those going on. Tonnage at the same time, overall tonnage of production has increased. In a couple years where tonnage was low yields, but now the bar is getting quite tight. The yields, the consistency is a lot better. Products perform in drier conditions, they perform in wetter conditions. The hybrids are just more of a manageable product. So that's acres and tons, and here's yield. So at the same time as the acres are going up, the yield's going up. So we were 25, kind of between 20 and 25 until 97. And then at that point we've started to increase, and the highest average bushel number for the Canadian production is just under 35 bushels. So the last few years the yields have been up considerably.
What would you consider seed production average?
Yields?
Yeah.
You're harvesting the female part of the field, so behind you, you'll get a look at it. You're probably 70% female, 30% male. So on that 70%, you can harvest anywhere, you can harvest 2,000 to 2,500 pounds of seed. So 40 to 50 bushels is a good yield in hybrids. And if you look at 70%, that would correlate to that area 60, 65, that would be the bushel you're taking off if you harvested 100% of the field. So the rates of pollination are good. There's definitely a time and place where 800 pounds, 1,000 pounds, 1,200 pounds would have been a hybrid seed field yield. But now they're consistently high performing at the seed level.
So I was trying to get my head wrapped around the significance of the economic impact of the seed industry here in Southern Alberta. Yeah. And I sat down with Troy Prasovsky. Sorry that he can't be here. He's with the Canola Council, but I think he's still in the hospital. His wife was trying to have a baby yesterday and it's not out yet. But anyways, we just guessed at, I don't know, 40 bushel average, but it's probably more like 30 bushel average from what he just said. And there's, 30 to 40,000 acres of seed production in Southern Alberta. We came up with a silly number like 500 billion at $9 a pound for seed. Like, have you guys ever thought is that
no,
right?
I haven't.
I checked my numbers; they were actually right, but that's just a tremendous amount of value. I mean, I know it certainly doesn't all stay here, but that's a huge thing to have in Southern Alberta.
Yeah, the investment costs. Southern Alberta is still a prime production area. As more commercial canola comes in, it pushes the canola because we need to have the isolations, we need to have the rotations, because if we get on too tight of a rotation, there's problems with volunteer canola, and that's a big issue today. So there is that pressure on the industry. Right now it's so invested in Southern Alberta, and it's still the best option for production. So that's why it's still here. That percentage could shift if other production regions open up, other irrigated areas, or if the tolerance to heat and drought gets better, you can move further south. Those are being explored in U.S. for the summer production, Chile for the contra season. But it is a huge investment, and it's a huge dollar volume that comes off the seed sales each year. So yeah, it's no quick changes are going to be happening at this point.
The Polish segment,
Yeah.
is that getting any larger?
I don't.
If we run into a year like this, it'll probably be pretty significant.
You know, last year everyone was looking for Polish canola, and you can't. It's not available. It wasn't available. In the end, we got a good October, and I think basically everything got harvested, and we're producing long season varieties. So, you know, this year we'll see what happens. Late seeding again. If we get a good October, if we harvest these late season varieties with June seeding again, then I think the pressure won't be there for these Polish canolas. But it's also the yield. The yield on this is 117% of Czech, and I can't tell you what Czech is, but it's not close to 35 bushels per acre.
That says the Polish Czech, though.
Yeah, exactly. That's the Polish Czech.
Well,
So
it depends on the years. Most years you don't need it. But
yeah, for sure. So, if you're seeding in June with a late season variety and you're harvesting it, then I don't think pressure will increase. Last year was the first year we were very late. This year's the second. If it goes fine, I think the talk will be less and less, personally.
The green count was terrible this year, though. I trucked lots into Blackie and Carsland, and
Yeah.
any green canola, they did not like you because of that. So
There's definitely green. Yeah, definitely
from
green.
that late harvest, right?
Yeah.
it's
So
regular canola though.
yeah, it's normal canola.
yeah,
The green count was terrible. Yeah, they combined them fine, but
Yeah.
there was just lots of green.
and on the
Trying to
commercials.
sell 12-13% green canola, it's a lot of fun.
No, fair enough. And what feed canola would go for, and what I don't even have any examples of what Polish canola yielded last year, or so how it compares. Even if it goes that feed level, speaking to the economics of it, I wouldn't be the best person to ask for that. But I haven't heard as much this year about Polish canola within the breeding and the production circles as you did last year. So we'll see. It's obviously popping up, but what it means going forward, I couldn't tell you that I think it would increase. But it's a very good question. It's valid because of the two late springs. Yeah, and I believe that's yeah. If anyone else have any questions at this point, that's
Getting back to the 1 mile radius,
yeah.
you're just trying to keep the bees in, or you think they could bring something in from the other field?
It's both. One, we want to focus all the bees in our field because it maximizes pollination. So that's why we don't bring bees in before the fields start to flower. Because if you do, they'll go anywhere else. They'll head to alfalfa, they'll head to neighboring canola, they'll head to weeds. And whether or not they're bringing in foreign pollen, you're getting less pollination in your field, so less return on the cost. And hives are a huge cost. They're our largest cost per acre right now in production. But yeah, the other is going and foraging in other fields and bringing pollen back to ours. Because your hybridity goes down, you don't make standards, the seed doesn't get sold, the farmer doesn't get paid, and everybody is unhappy. So it's twofold.
And it probably creates more ununiformity, if you will, in the seed itself. Like
Yep.
like
Yeah.
This gentleman's comment here.
Yeah, when you get crosses, you can end up with sterile plants in the hybrid, or you can end up with plants that are taller. The reason you pay so much money for the seed is because the expectation is you're going to get a tabletop, it's all going to start flowering the same day, it's going to finish the same day, and it's all going to look the same. So that is the standard that has to be met.
50 bushel if you pay that.
Absolutely, absolutely, yeah.
You managed, I was a bit late, you managed to cleaver problems all over starting up. So the big problem, clean the seeds.
Yep, purity. Seed purity is also an issue. Primary noxious weeds. Cleavers is a large problem. If you don't have a herbicide tolerant system, then you're not able to control your cleavers, and it's a seed similar size and shape to canola. So it's very difficult to clean out, almost impossible really. Wild mustard is another issue. So fields need to be free of those weeds. And also you do have certain isolation standards in certification, like 200 meters from wild mustard outside of your field. By certification, you have to have a half mile from canola. If it's not a half mile, your field will not be certified. So the industry standard's higher than that, but the minimum is a half mile.
And when it is a half mile, they use leaf cutter bees only, without the honey bees, because they don't have as big of a flying radius, so they won't go as far out of the field.
Yeah, and exceptions do happen because you do all your work to see what your neighbors are growing. It's something that farmers don't like to do all that much. You get involved in other people's businesses, but it is required to check your isolation. Because if you seed, you spray, canola pops up across the fence, then you need to figure out a solution to either have the neighbor take his canola out, or the seed field has to be taken out because it will not be certified. So at a half mile, like it was mentioned, the leaf cutter bees can be used, and they don't leave the fields. They're the bees that you see in these tents right here. And not all fields will have them, but when you have a short isolation, or if you're looking for dual pollination, you put them in. They stay in the field. Honeybees can fly vast distances.
How clean does their seed have to be? Like what's clean?
It has to meet #1 standards, but most companies go over above that. I think one of the philosophies is we don't produce weeds, we produce seed. So every company would have their own standards. They manage the cleaning, but I believe the goal would be 100% free of weeds, just as a broad statement. You're free to clean, you grade as you will, small seed, large seed. Seed consistency is important when you open a bag. You want to see consistent ball bearings in there, treated well and free of weeds. But personally, our motto is we don't produce weeds.
What's the percentage of off types that are in the seed?
For certification, the maximum number of off-type plants you can have is 1.5 in 10,000. So depending on genetic systems, previously the systems would have quite high frequency of off-types. Some systems, such as the MSL, do have generally a percentage of fertile plants in them that we send roguing crews through and pull those plants out. So at the same time, you can get a mix-up with your male into your female, something in the plant. The small contaminations do happen, and that's why we have crews of 30 or 40 people who come in early in flower and they pull any plant that's not correct. So we actually did find one here on the edge. Maybe we'll be able to find it right after. But after you spend a few hours in a field, you start to get the eye for what's not right and what is right. But for certification, 1.5 in 10,000 is what they inspect to. And
They'd all be rogue, like.
Yep. The idea is to have pure females. If your breeders and your parent seed programs go correctly, you won't have to rogue. But under certain systems, you do have to rogue a certain percentage of the plants.
So my name is Tina Hastings, and I work for Pioneer Hybrid, which is the plant right over there. I just graduated from university last year, so I have a master's in genetics. So I think that's why they asked me to do this presentation. But we'll get started. So I'm doing the science of hybrid production. And to start, who here has brown eyes? Anybody? Okay. And who here has a blue-eyed spouse? And who here has has blue-eyed children? Nobody has? Okay. So if you have brown eyes and your spouse has blue eyes and you have a blue-eyed child, you have one gene that codes for brown eyes and one gene that codes for blue eyes, and you can only donate half of your genes to your child. So your child has inherited the blue-eyed gene from you and the blue-eyed gene from your spouse. And your spouse only has the blue-eyed gene because the brown-eyed gene is dominant. So if you guys both have blue eyes and you have a brown-eyed child, I would check the eyes of the mailman. Start off with some modes of reproduction. We have self-pollination first, which is where the pollen from the plant would go back on itself, either on the same flower or the flowers below it, and it self-pollinates, and this leads to inbreeding or cloning. And then we have cross-pollination, where pollen from another plant will move to a different plant and pollinate it. So we'll start off with inbreds. An inbred is a population descended over several generations from a small set of ancestors and has a high rate of genetic similarity. So these are the clones. So if you look at this population here, they all look exactly the same. They all got the same genetics. They all end up being the same in the field. And this population here could be an open pollinated variety or the volunteers you find in your wheat field, stuff like that. And some of them are short and some of them are high and some of them have too many leaves and they just all look different. So the benefits of having an inbred population is you have uniformity and you have predictability, so you know what's going to happen. So how do we make an inbred population? And the easiest way to describe this is mass selection. That's the simplest method to do it. So with mass selection, you grow out your population and then you go through and you take out all the ones you don't like. So this guy's too tall, this guy's too short, this guy's too tall too. And then we allow the guys that are left to pollinate each other and grow and set seed. And then we harvest that seed, plant it again next year and do it all again. So plants are chosen according to preferred characteristics and allowed to pollinate and seed. And selection is repeated in a cyclic manner over several generations. So eventually, after a few times of doing this, we get a population where it's pretty much everything's the same. You might have to take out one or two, and eventually you get a population where everybody's exactly the same. So eventually this results in a uniform population. And this is the easiest methods, but we also have a pedigree method and a bulk population method and a single seed descent method. There's a whole bunch, but this is the easiest to understand. So why don't we just grow a bunch of inbreds since they're so easy to produce? And if we think about it, think about the genes. If all the capital letters here were favorable traits and all of my little letters here are lacking the trait, if we look at this individual, he has 2 copies of the capital letter for sclerotinia. So he's got really good sclerotinia resistance. And he's got 2 capital Bs, so he's got herbicide tolerance, so we'll say he's Roundup ready. But for these 2 genes, he's not very good with the weevils and he's kind of short, so we don't get as good of a yield. And this guy doesn't have any sclerotinia resistance or herbicide tolerance, but he's really great against the weevils, and he's really tall, so we get a better yield. Now, if we cross these 2 individuals, this individual is going to be able to donate these genes, and this individual is going to be able to donate these genes. So they each give half of their genes to the offspring with their cross-pollination. And we get a guy that looks like this. So this guy has sclerotinia resistance, he has herbicide tolerance, he's got weevil resistance, and he's got an increased height. So he's got a really good hybrid here. So why can't we make an inbred that has all the capital letters? Has everything. And for this, I'm going to use a stick analogy. This is really called recombination, but we'll just think of it as like a stick. So if you take a stick, and on each end of this stick is our genes that we want. So our herbicide tolerance and our increased height. And if you break it, you can break it in any spot. And then you can join those 2 other ends together and get the genes that we want on the same stick. And you're doing it randomly, you can't decide exactly where the stick is going to break, but it would be really easy to break that stick if they were on either end. Now, if the genes are right beside each other, right in the middle of that stick, and you randomly break it, it's going to be hard to break it in that exact spot in order to separate these genes. And a lot of the times, genes will slightly overlap each other, so you won't be able to break them apart at all. So sometimes the genes we really, really want are right next to or connected to genes that aren't so good. So this is where we come to our hybrid populations. These are the individuals that have 2 different sets of genetic instructions, but the population as a whole is identical. So each one of these guys has half of it donated by the mom, half of it donated by the dad. We got the right combination of genes, and they're all exactly the same throughout the whole population. So the benefits of this is we have uniformity and predictability and increased vigor, which is that heterosis that Ken was talking about. So heterosis, the best way I can describe it is that the whole is greater than the sum of the parts. So sometimes you get genes that when they interact together produce something better than either of those genes could do alone. So how do we make this hybrid? And we'll start off with the male flower. So this is our male flower, and it's a perfect flower. And we have the anthers that carry the pollen and the stigma and the pistil, and they're able to pollinate themselves. And then we have the female flowers that have lost their male parts. So either they're completely gone, the anthers are completely gone, or they're just little shriveled up nothings. So you plant them next to each other like in the field behind us, and then we put bees in, and the bees will go to the male flowers, get pollen all over their body, and then go to the female flowers, and the pollen is deposited, and the female is able to set seed. So why do we plant them like this? So we plant the male bays like in here, and the female bays right next to it. And the males will pollinate the females. And then once the flowering's done and we have all the seed we can get, we will mow out the males. The males have done their job and we don't need them anymore. And you can make what joke you want with that. So the seed from the female is pollinated by the male, and that's our hybrid seed. So we get rid of the males because the seed from the male was pollinated by other males, and that's an inbred. So we don't need that seed, we only want the hybrid seed.
So you're saying that's all we're good for?
Yeah, yeah. So why don't you, once you buy your commercial seed, you've grown it all out, you're taking it to the elevator, why don't you want to keep a little bit of that seed for next year and bin run it? And the reason for this is once you've grown out your commercial seed and it's a whole field of this, when they pollinate each other and pollinate themselves, all that recombination happens. And so if you can only donate, this is back to your blue-eyed thing, if you can only donate blue-eyed genes to your kids, that's all you're going to get. Whereas these guys have a blue eye and a brown eye gene, and they'll switch and mix with other genes. So if this guy donates these genes to an individual and these genes to an individual from someone else, now we have a plant that has no weevil resistance, and he's kind of short. And if he donates these genes to this guy and the recombination event happened and donated these genes, now we've lost our herbicide tolerance. So the field that you're going to plant the year after your bin run is going to be a dog's breakfast of all kinds of different plants. You're going to lose your herbicide tolerance in some of them. You're going to lose all sorts of things. So you've lost your uniformity, your predictability, and your vigor. And yeah, that's where I was going to end. So is there any questions?
You're going to have a heck of a time picking a husband.
Oh, thanks.
You know how to compliment a girl, don't you?
Yeah.
No one knows this, no way.
You got a buck.
She knows too much.
That's what I'm saying.
Now this year I heard that some bays were being mowed out early.
Some bays are cut early, and that's because the female is a little bit behind. So if you chop off the top of the male, they'll take a few days, but then they'll flower again, and so you'll lengthen the flowering time. So we're just, you trim some of the males every third bay usually, and then come back and do another third again, just to lengthen the time that the males flower, since they're so much more, they can pollinate themselves, so they lose flowers faster, whereas the female can only be pollinated by bees, right? So they are a little bit slower in their time. Yeah?
For pollination, or for a new seed, you need 1 year. It's possible to do it in greenhouse faster, like in the potatoes. They run all the whole year around.
Yeah, when you're doing the inbreds, yep. What they'll
or ship it to Chile or Argentina or
exactly.
Australia.
Yep. So to speed up the process, doing several generations instead of taking 7 years, you'll grow it out here, take all the ones out you don't want, send it to Chile or Argentina or a greenhouse even, and grow it out again, take out all the ones you don't want, send it back here, grow it out again, so you can do 2 generations in a year instead of just 1.
Yeah,
Exactly. Yeah.
Isn't there a great risk of bringing in that method?
Oh, with Chile?
The certification process in Chile is one of the main reasons seed is produced in Chile. because they have a really large export industry, everything from wine, but your fruits, your vegetables, fresh, right through seed. And it was really the corn seed that started the seed certification side of things going in Chile. It's the second largest producer of hybrid corn seed for the U.S., outside of the U.S. There's huge production list plants for Monsanto and Pioneer, but a large number of other companies, as well as European companies on the non-GMO sector. But SAG is their version of CFIA, and they inspect to Canadian standards. So they're trained by the Canadian, by the CFIA as to the standards, the inspection expectations, and so then their paperwork is acceptable for certification in Canada on our standards. So being weed free is critical. There are a couple weeds in Chile that you wouldn't find here in North America, and they're zero tolerance. Anything that has these weeds cannot be imported into Canada. So it has to be destroyed in Chile if it has those weeds that you cannot clean out. So this inspection and certification process is significant. The seed doesn't just get to Canada with no label and then get looked at. It's looked at right in the field, the same as it is here. But it's valid because there are weeds that we don't want coming back here. It's the same as clubroot disease that you'll get into with Rob. It's everywhere in the world, but we're doing everything we can to keep it out of Canada. That's why seed fields are all inspected for clubroot. If clubroot's found, there's an industry protocol for elimination, segregation and elimination of the infected areas. And so far, other than one potential hit that, it was unconfirmed in Jancia years ago, there has not been any clubroot found in canola hybrid fields. Southern Alberta. Clubroot fields.
How do I start? Probably I need to ask you something. Could you please get closer? And then I'm going to explain why. Probably it's very obvious, huh? I am not
Some of us don't hear that good.
That's the thing. I am not Canadian. My original language is Spanish, and if I try to yell, believe me, you're not going to understand nothing. Thank you very much for taking time. I know this is a busy time for every single person here, and I really appreciate you taking some time to come. This tells a lot to the Canadian and the seed industry. What's going on here is that there is interest. I mean, we were invited a month ago, say, hey, people from the industry, could you please make a module to talk about hybrid seed production? Say, whoa, this is something. So there's interest in what we do. It's not only production, but it's the seed business it is interesting. So we were very thrilled and say, yes, right ahead, we're going to participate. So the a number of companies came together. We thought, okay, how do we make a presentation that can bring knowledge and a little bit of interaction with the growers and industry? So these four little presentations were designed, lots of back and forth, what do I have to speak about, and so on and so forth. And my presentation is going to be based upon the field regulations and certification of hybrid seed canola. So I am I am what is called a senior agronomist. I work for Bayer Crop Science and for the bioscience group of Bayer Crop Science. And in essence, this group is the one who does the research and development, the production and marketing and so on and so forth of hybrid seed for Bayer Crop Science. When we talk about hybrid seed production, I mean, we're talking beyond what's happening in the field. It's not only the seed and the soil and the water. I mean, it goes beyond, because our industry, we have a framework that we have to work within. And this framework, it's quite strict, and it consists in a bunch of different norms and regulations that we have to comply with if we want to print a tag to sell seed to the marketplace. So it's That set of regulations, it's something that not only companies, but growers, seed growers have to deal with every single year. And that's, my friend Ken here is an expert in seeing what's happening sometimes when the inspection time comes in. It's a hectic time for everybody. So the Canadian certification system is one of the most comprehensive systems around the globe. It has been developed with the farming community and the industry in mind. So the Canadian government wants us to have the best possible material to grow a good crop. There's a lot of economic implications. We have to be efficient. We have to produce a lot to lower the cost and then penetrate international markets. That's one of the basis of a Canadian economy. So we have to be efficient. It's not only that we have the land and we have the water and we have the technology. We need to have the seed that is going to help us produce that kind of crops. So the Canadian certification system, beyond going and regulating our industry, meets international standards. There are international organizations, the OECD and so on and so forth, that tells this is the way how you have to produce seed if you want to trade it internationally. So the Canadian system meets the requirements internally and pushes up to meet international requirements. And therefore, our seed can be sold elsewhere around the globe. Our system is based upon 4 different components. There is the research and development, there's the registration, the field certification, and the seed grading system. I'm going to be briefly touching on 3 of these and a little bit more in depth in the field certification system. Research and development is what the guys, the researchers and brainies do at the labs. So they create those crossings, that technology that Tina was talking to you, they do that kind of stuff. They do crossings, they do lab work, they require pathologists and entomologists and geneticists and a bunch of stuff. And they create brand new germplasm based on old or new germplasm. So that's what they do. So this system, this subsystem, it's about just lab work and research, and they start documenting stuff. So when these guys have found there's a material that is interesting, that might help the industry, they move it to the registration portion. And then the registration portion say, okay, you have this, how do I know that this is what you're telling me? So this material it's taken all over the country for local and regional trials, looking for the expression of those specific traits, see how they behave. So some companies want to have taller plants, some companies want to have smaller plants. Some companies are looking for longer pods, more seeds per pod, for resistance to drought, name it. I mean, there are so many things that guys are looking for, but they have to prove the public in general that is what they have, and that's what the registration system does. They evaluate those hybrids, those materials in national trails, and they follow the documentation presented by the researchers. Fair enough. Okay, now we have a material that is promising to the market. Now, what's next with that material? Well, companies like the ones represented here, want to start producing those materials. So we select fields and we bring this parent seed to the field and we start just working this kind of fields. But those fields have to be inspected. Those fields need to comply with those norms and regulations. So there is the field certification system when there are very specific inspectors that have been accredited by CFIA that report to the CSGA come to the field. And they start looking for some specific things. They're still looking for the presence of noxious weeds. They're still looking for the presence of fertile plants on the female base. They look around to see if we were able to have the isolation, because the Canadian system says, you cannot grow a hybrid seed canola crop if you have anything in any other canola crop around 800 meters. So, and they start looking around, oh, you have one here that is less than 800 meters. That means that it doesn't matter what you did here. If you don't have the isolation, you're done. That field doesn't go for seed because we are violating one of the norms, which is the land requirements. So once the inspection is completed, the inspector comes and issues a crop certificate, which is a very nice paper that says, this land has the isolation, this land has the crop protection, those are the parent lines, male and female, used to create this hybrid. It's clean, then might be eligible to call it for seed, because it only becomes seed when it's being graded at the plant after harvesting. So before that, it's just a crop. It's just a crop. We are intending to produce seed, but the grading system is the one that is going to tell us, yes, it is seed. And how the grading system does it? Well, you all know there's grading tables. There are grading tables, and there are grading tables for hybrid seed canola. So the grading system says you need to have certain purity. You need to have a number of green seeds on that crop. No more than this. This is the threshold. You need to have this germination percentage, this percent of germination, and if all is good, then the system is okay. You can print the tag, and then you can start working on those bags. In Canada, there are two groups working on this development. So we have the private sector, again represented by companies like Hightech and Monsanto and Dow and Bayer, and well, there is a number of companies, and that is the private sector. And we also have the public sector represented by the government and its organizations like Agriculture and Agri-Food Canada and universities. In essence, both groups do the same thing. We're looking for the best germoplasm. We're looking for the breakthrough that's going to bring a lot of yield with the minimum input to the grower. The main difference here is the funding, where the money is coming from. Because developing a hybrid line is extremely expensive. It takes years and years and years of investment and research. I mean, I'm not lying to you if I tell you that every company can test 15 combinations every year looking for one. And that is a lot of brain power. So lab work and so on and so forth. It's money that comes into this that has to be, accounted for somewhat. So the researchers develop this germplasm. When they feel the germplasm is good enough, they move to the trials that we just spoke about. The Canadian system is very strong, and the Canadian seed is very strong. And one of the reasons why it is so strong is because in Canada, we have the plant breeders' rights. And this is a piece of legislation that is within the Seeds Act that protects the researcher and protects the company that the researcher is working for, protects that intellectual property and that investment. Because, as I said before, there's a lot of money going into this. If I create a very specific hybrid, I want to have control over it. So those plant breeder rights protects that intellectual property, and protects it not only for my investment, it protects it for the Canadian industry. Because without having that protection, there is no way nobody's going to invest if they don't have an insurance on that investment. So this is the insurance. If the seed that, as I mentioned before, goes elsewhere in the world, different people in different countries may misuse that seed. So there is a system that protects the misuse of seed. And that is why there is investment and there's development, and the people like to do the research on seed production, on new hybrids and new germplasms in Canada. Moving into what it is now, the seed production itself. If you go to any dealer or you pick up the magazines, companies advertise the seed. But no seed can be advertised in Canada if they don't belong to a pedigree background. So there should be a background. I mean, this seed has to come somewhere, because I can't just bring it back to you and say, hey, there's a good seed. Oh, where's it coming from? Well, it's a seed, yeah. So, but every single person, every single company has to provide that background where the seed is coming from. And that's what is called the pedigree. So it's a trace back from the origins of the seed. The system is based upon the regulations of the Canadian Seed Growers Organization, and the members of that organization, they provide support, they provide documentation to say, okay, this hybrid 123 has their parent line ABC and XYZ. So there is a background that produced this hybrid 123. There are different classes of pedigreed seed. Okay. The highest, or lowest, depending on how you see it, is the breeders. So the breeders are these guys that I said, small plots, cages and labs, and they do the crosses that Tina showed you. They can have multiple generations. But once those researchers released the breeder seed, there can be only one generation of foundation. There is no F2 on foundation, there's only one foundation. And then there's going to be only one generation of certified. So there is no F2 for certified. So it moves from, can be multiple generations here to one, and then it goes commercial. It can be regrown. And it can be regrown, I mean, same thing, because what Tina explained. I mean, there's breakdowns, there is things that happen with the system, with the seed, that it just loses their capacity to maintain the vigor. That is what makes it so special. All those regulations and all those norms, are in the Seeds Act and within the Circular 6 of the CSGA, the Canadian Seed Growers Organization, and the subsection 5 is the one that deals exclusively about the production of hybrid seed canola. It's a little paper, it's not too complex, but it's good to know what the regulation says. Because, as I said before, those regulations are, the way how we manage fields is not because it's an arbitrary decision. It's not because I want to have this or I want to have that. That is law, we have to abide by that. And that is the document that tells us how to do things. When we talk about pedigreed seed production, there's 3 stages. The production itself, so we have the seeding, irrigation, and plant management, and pest control, harvesting, plant nutrition. That is one of the stages, just production itself. Then, once the crop has been produced, what happens with that crop? The crop has to be managed in some way that we can keep the integrity of that seed intact. Because we're working with living organisms, and that life and that strength has to be passed over winter to the next spring. So those materials have to be very carefully treated, very carefully managed, I should have said. So if you're a seed producer and your agronomist is coming to you before harvesting, likely he's going to tell you, you know what, I want to inspect your harvester. I want to see that it's spotless and it's perfectly clean. Because if that harvester was used in a previous canola crop, there might be foreign material in that harvester that can contaminate your seed. So that is not good. Your hybrid was good. You're going to throw it just right down to the trash. And the same thing happens with the transportation equipment. The trucks have to be clean. And the bins, when the seed is going to be stored, they have to be perfectly clean. And more importantly, once the seed is going into the storage, those bins are weekly monitored. So we have temperature control sensors, and there is oftentimes blowers and dryers and all that stuff, because we want to keep that seed in the best possible condition before it goes to the plant. Because then when the seed hits the plant, oftentimes before hitting the plant, it's tested for the grading system that after it's going to tell us, yes, you can print the tag, you can put that tag in the bag, The bag will go to market. Before that, there is no seed. There is no seed. Even if you have a perfect clean crop in your bin, it comes to the plant, and the grader says, you have cleavers here. It's a noxious weed. Doesn't matter what you did before. Noxious weed, done. Ciao.
Do you size the seed?
Do I size the seed?
Is the seed sized at all? Like the smaller kernels out, or?
Yes. Yes, indeed. That happens. I mean, there are different sizes of sieves that are used when the seed is being cleaned. And usually the smaller seed is thrown out. And because it's the way how the cleaning system works. Okay, and this is probably this is my last board here. These are the things that require the law. The law requires us to comply with. And the land requirements. So we want to have fields that have not been that haven't been grown with canola the previous 4 years. So we need to have at least a 4-year rotation. And even if we have the 4-year rotation after canola, what's going to happen is the inspector, the agronomist, is going to come to the field the year before sowing the field. He's going to walk it, and if he sees volunteer plants and still in that field, even after 4 years, he's going to suggest to the grower, it's not a good idea. It's not a good idea because you can have volunteer plants in your female base, and then that's going to trigger rogging. And if the population is too high, it might be unroggable. And if it's unroggable, when the inspector comes, say, oh, Jesus Christ, I mean, this is full of volunteers. You have a very high risk of cross-pollination between your volunteers and your canola. So I am sorry, this field is not going to pass the inspection, and unfortunately, it's not going to make it for seed. So that is very important to have that rotation. I also mentioned that we need to have a pedigree background. So no seed different than breeder seed or foundation seed can be used to produce certified seed. That's a given. So it's either foundation or breeder seed to produce certified. If we don't have any of those, then we cannot produce seed. The crop inspection, the inspector comes at blooming time, just a little bit after this, and they're going to look for the overall health of the crop. They want to see the male and the female base. If the male base are gone and you have only females, they can expect that's gone. So it has to be before mowing. So and they check for everything in the field. They check for the isolation. They check for the isolation. There's the seed requirements. In Canada, this is an interesting point. In Canada, we cannot produce a hybrid canola field if we have around the 800 meters radius and our canola crop. If we don't have those 800 meters, then the field is not going to be accepted. But even if we have the 800 meters and there is canola around, the inspector is going to tell you have to rogue that canola at 300 meters because that canola cross-pollinates. If you don't clean it, if you don't rogue it, it's not going to pass the inspection. It's not going to go for seed.
Are they not changing the crop inspection procedures like with how the CFIA is executing it now? Isn't that being changed? Is it still the same?
No, it's still the same. That still is the same. Well, there are some talks right now on how to do the inspections. Apparently some companies are going to be allowed to do their own inspections instead of third parties, but that's still a long ways to get there. But I mean, there might be some changes on that. So once the field has been inspected, the CAGA releases the crop inspection report. Says, yeah, it might be used as seed. They make sure that with their agronomists that the seed equipment that was cleaned before seeding, that the storages were clean at the time of storaging, and the seed was graded. If we met all this stuff, then they're going to allow us to print tags, fill the bags with the seed, treated, fill the bags, and send it to the marketplace. That's in essence how the system works. And I don't know if you have any questions, I'll be happy to answer. If not, that is the
There's one here.
yeah, Chris.
It seems like the hybrids have a longer dormancy.
Mm-hmm.
I mean, let's see, you have a canola this year, and there's canola plant showing up 4 years later. You think you have a good herbicide program to take out volunteer canola and succeeding crops. It seems like the hybrids, 4 years later, you still see.
Yeah, well, it's not a problem of dormancy, honestly. I think it's more a problem of crop management, land management. We've seen situations very, very specific with potatoes. I mean, because if you grow canola and the year after you grow potatoes, when you're preparing the land and making those deep beds, essentially what you're doing is you're burying the seed. And every year when you come and prepare the land again, you're going to move in seed up and up and up and up. So that's why we very strongly require that the year after canola, just try to make a light preparation. Don't bury those seeds, because there will be seeds in the field. There will be seeds throwing out of the combine. Seeds are coming just drop out of the pods. So just a light preparation and good 2 or 3 herbicides application after canola. That's going to ensure that you're going to start reducing your volunteer population within the next 4 years. I mean, the companies right now are very concerned in that specific fashion, how to make sure we can grow canola 4 years after. Because the amount of seed the industry is blooming, it's going just nuts. So if we don't manage that canola land in order to make sure that we can meet it at 4 years, then we're going to go every 5 or every 6 years. And then at one point we're going to run out of land to grow seed canola. And oftentimes that has happened. That oftentimes, I mean, there is nowhere. I mean, yeah, we have the 4-year rotation, but I still have volunteers. So companies are very concerned how to manage land to make sure we can meet the 4-year requirements.
Yes, I'm Rob Waters. I'm with Monsanto, and yeah, as I've been telling the previous groups, I'm a little bit sheepish about speaking here today, because I can see there's a number of people in the crowd who are at least as knowledgeable about hybrid canola seed production as me, and maybe even more. So we'll keep it interactive, and if I say something that doesn't sound right, or you say, gosh, that's not the way I understand it or know it, please speak up, because I see this as kind of a sharing session here. So disease control and hybrid canola seed production. So that's one of the things they wanted me to talk about. And I think really for compared to a commercial crop, so I'm gonna kind of talk down to a level of a kind of commercial grower. I think in a commercial crop, really it's very similar in a lot of ways to what you'd see in commercial canola, especially for the first two I'm going to talk about here. So seedling disease complex, that's kind of groups together root rot and brown girdling root rot and all these different kinds of things that we talk about as seedling diseases. Damping off, this is a nice little damping off picture. If you ever pull up a seed, dig up a seed out of the ground and you get that constricted root there, if you can see that, nice fat fleshy one, and this one's kind of constricted, that's damping off. The cure for that is treated seed, right? So if the seed treatment's working effectively and you don't get a year, a spring like this year where it rains every 3 days and the treatment wears off eventually, right? But the cure for seedling disease complex that we know so far is treating the seed. Sclerotinia is another thing we worry about in hybrid canola seed production, and I think more and more for commercial seed growers, I can see this becoming more and more of a concern as well, mainly because of rotation shortening, right? So as your rotation shortens, you've got a higher chance of having sclerotinia in your seed, right? Or in your commercial crop as well. And we worry about it in seed too. I think the disease cycle is the same, whether it's commercial or a hybrid canola seed crop. Starts with the sclerotia body inside of the host crop from the previous year. This is a canola stem, but it could be beans, it could be sunflowers are very susceptible to sclerotinia. This hard little body turns into this little tiny mushroom, and it's very small. I got to be honest with you, I'm not 100% sure I've ever seen an apothecia, which is this little mushroom here. It's tiny. Like, can you see the guy's handprint behind it? So I've had growers come to me and they come to me with this mushroom that's about this big and they say, oh, here, I got sclerotinia in my field, I got this little mushroom. No, this thing's tiny, right?
They also only come out for about 2 days.
Yeah, like I said, I'm going to sound like a terrible agronomist, but I'm not 100% sure I've ever actually seen one. And I think if you did, you need to have your butt up in the air and your face down on the ground to find it, right? If you're walking along and you see it, it probably isn't the apothecia, right? Yeah, what happens is the spores shoot off from this little mushroom. They land on the petals of the flowers. So this is how the infection has to occur. It lands on the petals. The petals eventually start to drop off, and they fall into what's called the leaf axis. So that's anywhere where the stem or a leaf is coming off there. And that's where the infection occurs. So the spores onto the petals down into the leaf axis, and that's where you get the infection. The cure for it is to spray fungicide. Fungicide needs to be sprayed when it's flowering. If the plant isn't flowering, or if it's out of flower and you're seeing this, you can spray your brains out, but it isn't going to kill anything, right? You're too late. By the time you see the disease, you're too late to spray it. This crop here, if it gains 5% flower a day, I'd guess in another 3-4 days you might be at 20% flower, you could spray it, just as a bit of a guideline. Obviously there's two different things going on here, right? But yeah, exactly. I'd say the male is probably almost there. You're at the early stage of it. First petal drop is something to look for. And I don't quite see it yet. It's really close. It's really close. What's that? What percent?
Watch for that when the petals start to drop.
Yeah, absolutely. The first petal drop. That's the thing I always look for when I try and make a recommendation. And then that gives you a window. I always say you're gaining 5% flower a day. So if you look at it today and say, oh, this is 10%, when am I going to go? I'd say 2 days from now, 3 days from now, you better be getting that plane ready or your ground rig or whatever you got, right? Yeah, absolutely. The male always looks a bit yellower too, and I'll explain that. But
I'm going to have a problem this year, though, no?
Sclerotinia is the funniest disease. It is the strangest disease. I've had years in fields that didn't have crop history of Sclerotinia or Sclerotinia tolerant susceptible species in it, and that have been loaded with it. I've had fields 2 miles down the road that were dry beans every second year for 30 years and had nothing. I'm not kidding you. I mean, same climate, it's the strangest disease. I think it's a fool's game predicting the Sclerotinia infection. The weather, so I'm going to counteract all the things that you read in the publications. Look at the weather, look at the disease triangle, look at this. Yeah, that'll help, but you can get hit. That's my message here, is that you can watch that nice disease triangle, have the conditions and the rotation and everything, and not have any infection. You can have conditions that you say it won't be bad, and you get loaded with it. So it's a crazy disease. It's a tough one to predict. I would say that we're getting into a day and age. In seed production, it's kind of a no-brainer because it's such a valuable crop, right? But I think we're getting to the day and age in canola here where the canola price is high enough. It's an expensive spray, but what we used to say, what I've been told, is that if you pull the number of plants that have infection, so when you get to this stage, it's too late to spray, but if you want to know how much you lost to Sclerotinia, just stop in a row somewhere, pull 10 plants, the number that are infected, divide it by 2, and that's your yield loss. There's a lot of buzz about clubroot probably starting back in about 2002 when the first fields started to be identified up in that kind of real hotbed in Sturgeon Valley, Sturgeon County. So what happened at the time, and I think this is kind of the most interesting thing in terms of disease management and hybrid canola seed production, is the companies got together. So really from feedback from customers. So customers came to the companies and said, gosh, we hear about seed production and in Southern Alberta we hear that there's a field that was found with clubroot. What are you companies doing to make sure that when I buy that nice bag of canola seed that I'm not getting clubroot coming in with it? Clubroot is primarily spread by soil. It's a soil-borne disease primarily. So the biggest concern is when you have soil movement, right? But in theory, it could be moved on seed, right? Seed isn't 100% clean of every particle of dust, of course, right? And so in theory, it could be moved on seed. It's highly unlikely, but that's not the way the companies looked at it. They said, look, if it's a concern for our customers, then let's do something about it, right? And let's have a collaborative effort. Let's have a common protocol so that all of us can go to the industry and say, no matter what bag of canola seed you buy from what company in Canada, you're going to be assured that those fields are scouted for clubroot and that we're reasonably sure that you're not buying clubroot, right? So what we do is the best time to scout for clubroot is really after flowering in pod fill, and that's why these pictures are down here. What happens in these canola fields that's kind of unique is that once the female has been pollinated by the males are destroyed, right? They're mowed out, they're destroyed. So this is like a mini production field here. Every 20 to 30 to 10 feet, depending on the field and the system, you've got this nice row running down the field. So you can picture the intensity of the scouting that you can do in a field like that, right? I would argue that in terms of the intensity of scouting for disease, it's probably the most intense disease scouting that's done in any crop in Western Canada. outside of maybe potatoes. Maybe a potato guy might give me a run on that, but I would say hybrid canola seed production fields are scouted for clubroot at an intensity that no other crop is scouted. So I say that a bit brashly just to convince people that there's something being done, and on top of that, in the 4 years that we've been doing it, not one field has ever been found in Southern Alberta with clubroot in it in a hybrid seed production field. So I don't want to say it could never happen. I don't want to say that we've got a foolproof method here, but it's definitely was taken serious, and I think the proof is in the pudding that we're doing intensive scouting and they're not finding it. So, okay.
What would you be looking for?
Yeah, okay. Dead plants. So the dead giveaway, and the protocol was actually developed with just with the companies. We actually, Alberta Agriculture, Murray Hartman was who helped us develop this protocol. So we didn't make this up on our own. You're looking for dead patches. It looks like sclerotinia infection, in fact. But the dead giveaway is you pull it out and those nodules on the root. So that's what we train the scouts to do. If you see a dead patch, when you're driving down, you can go down with a quad, you can walk down. It's at a certain intensity throughout the field. You see a dead patch, pull up the roots. If the roots have lumps on it, then we start taking samples to labs to ensure that that's what it is, right?
I just thought that was the new nitrogen fixing.
Yeah, exactly. The nodules on the root there, right? You never know, the day might come, right? But anyhow, so that's what's being done about clubroot. I'm going to hand these around. These are some female flowers. So for anybody who's never doesn't know what the difference between a male and a female flower is, I want you to pull one of these flowers and then pass it on to the next guy. And try and pull one near the top. They tend to be a little bit more open and easier to see. So pull one near the top, and then I'm going to go grab some male flowers here quickly. So the essence of seed production, of hybrid seed production, is really the crossing of the male and the female plants, right? And so you can clearly see what we've got here is we've seeded sort of a little mock field here, right? So this is a and it's the bays are right. The bays are the right size, but normally you'd see this if you drive around Southern Alberta in the summertime. You're going to see these quarter section fields of male and female plants, and they'll have this leaf cutter tent in the middle of it, right? And really, I'm going to start this one. This is our male plants here. Again, do the same thing. Pull one flower, pass it on. And essentially, the essence of what we're doing here is crossing two distinct lines, right? It's almost two different varieties, two inbreds as they would call them, right? And I forgot to pull my thing here. One's a male and one's a female. So the male crosses with the female, and that gives you your F1 seed or your hybrid. This is the stuff that you're buying, right? In the bag from one of the companies. Now, you might be driving down the highway and you see one of these fields and you say, boy, that male is really tall, or that female is really short, or that thing lodged bad, or it looks kind of funny, or gosh, those female flowers aren't very bright. That doesn't look like I want to find out what variety is. I don't want to buy that one, right? It's no good, right? There's something wrong with that stuff, right? No, right? That's the quirk of hybrid seed production. It doesn't matter whether it's corn hybrid seed production or tomato hybrid seed production or canola hybrid seed production. It isn't necessarily that the male and female pass on every single trait to the F1. So if you see a seed production field that looks kind of wonky, it doesn't mean that the F1 that you're buying is bad. In fact, what a plant breeder will tell you, and if you talk to corn breeders, they'll tell you that it's corn breeding, corn hybrid canola seed breeding is very mature. They'll tell you that the wonkier the inbreds, the better the hybrid is. The more crazy, the more those cobs will look diseased, they're poorly filled, and yet that's what makes the best hybrid. Right? The wonkier they are, the better the hybrid. So don't judge a book by its cover is what I'm trying to say. If you're driving down the highway and you see something that looks kind of crazy. Okay, male and female flowers. So if you hold up those two flowers that I gave you there, that you got, did everybody get a chance? So you'll see one is probably smaller than the other. The female is typically a bit smaller, and that's why the female doesn't look as yellow. So if you try and judge the percent flower on these fields by the yellow content, you could be off, right? Because I think the male is a bit ahead on the female on this one, but you can look at a field and have it like this, and you can say, oh, the female is way ahead of the male. Not necessarily. The flowers are just smaller, right? But if you hold it up to the light, what you'll see on the male, the bigger one, is it looks like it's got a few extra parts. It's bigger, and you can see that it's got these things kind of sticking up. And the best way I can explain it is it's got like the pronghorn antelope horn hook on it. Can you see that? You kind of might have to turn it a little bit, but it's got a nice little pronghorn antelope hook on it. That's the anther. That's the male part of the flower. We call it a male flower, but really this isn't just like conventional canola, commercial canola that you would grow in your field that you'd buy. It's a male flower, but it's like conventional commercial canola, right? The one with the trick is really the female, right? It's sterile. It has no anthers on it, or it does, if it does have anthers, you'd have to peel away the petals to see them, and they're really shrunk up. They're down deep in there and there's no pollen on them. The other dead giveaway is if you rub that male flower, you should be able to get some pollen on your fingers. Today's a good day. Sometimes if it's really wet, if the pivot's gone over, or if it's a dewy morning, you won't get any pollen, but you should be able to rub that male flower and get pollen. You should be able to rub the female flower and no pollen. So the pronghorn antelope hook and the pollen on your fingers will tell you male from a female, right? So, got a male and a female here. Why do we need these bees, right? It's windy in Southern Alberta. Wind must move the pollen around. Why do we need a vector to move it? It sounds a little weird, but canola pollen is actually quite heavy, and so it doesn't move readily. It'll move within the first 2 or 3 feet, so those first few feet of the female bay might get pollinated quite well. But if you want to get it all the way through the bay, you need a vector, and the best thing that we've found are bees. 2 kinds of bees that are typically used. We use honeybees. They're placed in the corners of the fields, on the pivot corners, mainly because honeybees will fly further. They don't need to be directly in the field. And we use leafcutter bees, which we've got right here. And we've actually put some, if you see some things flying around there, those are the leafcutters. I brought a few of them out here yesterday. They're actually flying around. Don't worry, they're not particularly dangerous. They're not particularly aggressive. They don't sting like a honeybee does. You don't get anaphylactic shock from a honeybee or a leafcutter bite. It's a physical bite. If they do bite you, they might get caught in your collar and bite you, and it'll hurt, but it's nobody in the history of the world ever died from a leafcutter, right? As opposed to a honeybee. Yeah, so that's the vector to move the pollen from the male to the female, right? Why do we use both? Why don't we just use, which one's better, right? Leafcutters or honeybees? Leafcutters work in, this is beautiful leafcutter weather. Perfect, right? Hot, no wind. These guys are loving it, right? So on a day like today, the leafcutters, I hope I don't offend any honeybee guy here. The leafcutters, I think, will out-pollinate a honeybee. So why do you want honeybees then, right? If it's cool, if it's wet, if it's windy, if it's rainy, the honeybees, I think, in my opinion, will out-pollinate a leafcutter. So really, the reason why you use both is a bit of a hedge against the weather. You might use exclusively leafcutters, and the reason why they're placed in the field, because they don't fly very far. So honeybees will fly a long ways, but a leafcutter stays pretty tight into the tent, right? And so you'll see them spaced throughout the field about that increment, because they'll only fly that far from the shelter, right? So, the other point I was going to make here is essentially often we're really growing 2 different crops here, right? 2 different varieties. So, Kent, if this was your field and you looked at those males at about this time of year, what would you be thinking of doing? Yeah, so Kent's an experienced hybrid grower, and he looks at that and he says, geez, those males are ahead of the females. And I always compare it to cattle, right? If you had a pasture full of cows and there's no bull in there, what kind of calf crop are you going to get? Not a very good one, right? And that's the same thing here, right? These females have no ability to produce seed. So what Kent's seeing is he's saying, geez, I could see those males coming out of flower before the female does, and that's a problem, right? You're going to have cows and no bull. So when you go in there with a mower, just an old rotary mower or a flail mower, and you chop off the top 6 inches, 8 inches, 12 inches. It'll depend on the hybrid, and they're all different, right? Some have the females way ahead, sometimes the males are a little bit ahead, sometimes the females are ahead of the male. So you cut the female back, which is a bigger problem, of course, because it's a wider bay, right? But there's ways to do that as well. The other thing I was going to point out here, so I showed you a male and female flower here. So Jack Van Trey, when you look down that female bay, what do you see? I can see him.
I'd pull my agronomist.
Yeah, and say, what you knuckleheads, you gave me some seed that's got fertiles in it, right? So you can see them. Now that I've shown you the male from the female, you can see some bigger flowers in there. Now that isn't from, this female isn't contaminated. What happened was they didn't clean out the drill good enough. So they were using the same drill to see the males and the females here, and you can see the males popping up in there. And when they send rogers through, oftentimes it isn't because the grower didn't clean out the drill well enough. It's volunteer canola, like we were talking about. Often it's volunteer canola. And that's what the rogers are looking for, right? They're looking for that bigger flower, and you can see them. It's hard to believe, but until you show somebody it, if you see those bigger flowers out there, those are males that are in the female, and they have to go, right? We don't want that harvested with the female. We don't want the male seed. We don't want the female seed. We want the cross between them, right? But so
we're all the same, eh?
yeah, exactly. But I think, I was going to pass around this. So the leaf cutters here, they're they overwinter as essentially as a pupae, right? So these little the nests that they built here, these little pupae that they've constructed, are made out of leaf material. I'm going to pass this around. There's no bees in here, no live ones at least anyways, they've all hatched already. So we bring them out. This tray would normally be full, and you probably have 2 or 3 of them per tent. So that's just a few of them there. They overwinter them as the pupae there. They bring them out, they incubate them for about a month, 30 to 40 days. So 30, 40 days before this crop starts to flower, we have to start the leafcutter guy to incubate them, right? We bring them out as they're hatching. They place them in the tents, they hatch. You can see them flying around there. They pollinate the crop, but at the same time, they're cutting pieces of leaf material. They're collecting pollen and nectar to use as a protein source to lay eggs. They lay eggs inside those blocks there, those styrofoam blocks. At the end of the season, essentially the leafcutter keeper harvests the bees, right? So we don't get the bees back that are already hatched and flying. They die, but the eggs have been laid in those cells, and as is being passed around there, those are pushed out. Essentially, there's a machine with fingers that kind of pushes them out of the blocks, and then they're stored in cool, dry storage and ready to be incubated the next year. So that's the cycle with the leaf cutters. So I think I've used up my time here, probably more than my time. But are there any kind of quick questions here about hybrid canola seed production?
You guys, I know there was a trend, narrow bay production. I know I horsed around with that for a while. It's kind of a pain. You ride the bays on a lawnmower, basically.
Yeah. So if you have a look at this here, can you see what's going on here? So as compared to this or compared to what's behind us here. So there are some companies that are doing row crop production. And there's a number of different reasons for that. And all systems are different. Bayer's system is different from the system that Monsanto is using, is different from the system that Dow might be using, is different. So all the systems are different. All these sterility systems aren't the same. They aren't all common, right? Sometimes you need to have the females closer to the male, right? And that's what's happening here, right? They've got 4 rows of female and a row of males. So right, this whole, from the male to the female in this system, the male's here, the little 4 rows of female 22-inch centers, next male's right here, right? They do these things for a reason, right? Some systems need the males really close. Other systems, like this one, can do it further away, right? And that's why. And there's a secondary reason too, that you might do this type of production, this row crop production. It's you can inter-row cultivate for volunteer control. So you don't have to rely completely on the chemistry. So that's another reason why you might see a bit of more of that happening in the country as well.